Hello, I'm Ethan Macklem, and my project is over the usage of Nasonov gland pheromones to lure Aphis mellifera to struggling plants. So, Aphis mellifera, also known as the Western honeybee, is a eusocial organism that basically works with the other organisms in the hive to create almost a superorganism that works together completely. Also, the primary role or most common role for honeybees are pollinators, and so this also helps with biodiversity as they go from plant to plant and help pollinate various different types of plants. Honeybees also commonly communicate through pheromones, which are chemicals that allow them to disclose information to each other. So, as previously stated, the bulk of what we're studying is the Nasonov gland pheromones. So, a little bit about the anatomy. The Nasonov gland is located in the groove between the sixth and seventh targites. And so, to release these pheromones, they expose the groove, and this will lead to the secretion of the Nasonov gland pheromones. And as you can see in figure 2, which shows the anatomical locations of various different hormone releasers, you can see that the Nasonov gland is at the rear end of the bee. Next, we have the Nasonov gland pheromone function. So the pheromone is primarily a worker-only pheromone, and it helps to orient other bees to food as well as nesting sites. And they also commonly mark hive entrances and food sources with the pheromone so other bees can remember. And additionally, it's commonly used for long distance communication. And so figure 3 shows the pheromone pathway for many different pheromones, and it goes through the process of the antenna as well as the brain. But the primary question that we're trying to focus on here is, can Nasonov gland pheromones lure Aphis mellifera to struggling plants? And so a little bit of the research findings. So in a previous study, they examined the use of Nasonov gland pheromones to treatment on pear plants, and they basically characterized this using honeybee abundance as well as the flower visitations. Additionally, these experiments were conducted at the tree level and the orchard level. So the tree level was about like 6 trees that were individually treated, and they studied the honeybee abundance and visits, whereas orchard level was the whole orchard, and they compared that to one orchard to another that was treated and untreated. And so the results show a significant increase in both the honeybee abundance and visits at both the tree and orchard levels. So on the right-hand side, you can see in figure 4, there's a significant increase in the number of honeybee abundance and visits when compared to the NGP lured plants and without NGP lured plants. Additionally, you can see in the part that is labeled with absent, so that just talks about the orchards and trees that didn't have a hive nearby. So you really don't expect much change if there's not a whole lot of bees nearby. From that same study, they did a wide tube olfactor meter experiment. And so they performed it using to test honeybee preferences to NGP. So one side of the Y tube led to NGP and the other side did not, which is the control. And so bees were placed in the middle chamber of the Y tube and allowed to choose which path they wanted to go. And so in an experiment of 91 bees, a significant number of them chose the NGP path. And so on the right side in figure 5, you can see from the graph, a total of 68 of those 91 bees chose the NGP, whereas only 23 chose the control. And so that's a very significant difference, and that just demonstrates how much of a preference these bees have for NGP. Next, the last study that we'll look at is very similar to the first one, and that it looked at the number of pollinators present in um, around plants. However, this study, as opposed to the other one, this one studies blueberries, the other one studied pears. And additionally, this one studies different types of bees. However, there is still Aphis mellifera data in here for honeybees. And so this study found no significant difference in the number of pollinators present for blueberry plants and blueberry plants treated with NGP compared to untreated. However, if you do go in and look at the data here, you can see an upward trend in almost all of the honeybee data. So for the near, intermediate, far, and central, if you look, you see a positive trend in the full pollinate compared to untreated. And full pollinate is basically just the same as NGP. So conclusions here, this evidence supports the hypothesis that NGP has the potential to lure Aphis mellifera to struggling plants. So in experiments of real world conditions, such as a full orchard, a couple of trees, as well as in-lab experiments, they both demonstrate how honeybees are attracted to NGP. And lastly, these findings can be advantageous to farmers who need a stable pollinator and lack such. So the broader significance of these findings are that Nasanov gland pheromones can be utilized to help mark struggling crops that lack pollination and attract these Aphis mellifera honeybees who can assist in pollinating them. So they can more widespread, more um, thinking forward, you may say. This can help assist in growing crops in more remote locations where pollinators are sparse and not as common. And so you can present those pollinators there, and you can also mark these plants with NGP and hope that the pollinators will go because they have a preference for that. So here are my references, and thank you.